Thanks very much, John. Yeah, thanks to the organizers for having me here. I'm very honored to give the closing talk here. Quite daunted because I'm neither a structural biologist nor a developer, and so bear that in mind, please. No, so instead, I basically take all of your hard work and I try and make the structures that you produce as beautiful as we possibly can. And so I do that with our small animation company in London called Phospho. And I can explain a lot of things about what we do, but the best thing is to just have a look at some of our work. So I've got our general kind of showreel, which has some non-structural things in there as well to start with, and then afterwards we're going to dive into the structures. Yeah, like John said, I'm a kinase guy, so I had to obviously close that one with the kinase. And so we're not animating. We like to do illustrations as well, and there's, like John mentioned, we like to try and get journal covers, and we've got a pretty decent track record of getting structures on the cover of journals as well. So one of the things that I wanted to talk to you about today is the type of challenges that we tend to run into when we want to visualize protein structures. And I think a lot of these things can be transferred to kind of the sorts of decisions that you run into when you're making your structures for like preparing your paper figures and presentations and things like that. And so the three things that I wanted to talk about in that sense were the kind of some of the challenge about showing your structures clearly, about showing 3D structures in two-dimensional images, and how you can show dynamic processes. So to start off with a very, very old picture of mine from when I was still in the lab. This, don't be fooled by the 2018, it took years to get this out. So this is a 2 or 3 interaction that I was working on, and an image that I originally made in PyMOL. And I've never been a big fan of how PyMOL renders things, and so some of the things that I think can be quite jarring when you're looking at a structure, and particularly if you're not a structural biologist and you're trying to navigate what it is that you're looking at. So these things like the sort of harsh shadows that you can see in the top right here, and these sort of fake reflections where you don't actually have a light, but there is a reflection there somehow. I think these can kind of take away some of the emphasis of the actual protein structure that you're showing. So at the bottom here, I've got, and actually, sorry, as a caveat, I know that that image was made in something like 2014, and I know that in the meantime, there have been all these great advances, and we saw yesterday in Anna's talk that in Qt you can do amazing ambient occlusion and things like that. And so it's looking a lot better than it did, definitely. But at the bottom is what we ended up going with in the paper, and so this is how a kind of a clean render, how we would do it. And so the rendering engine here, we took the protein structure out of Chimera, put it into our animation software, and then we rendered it using our favorite rendering engine, which is Pixar's RenderMan. It's the software that Pixar themselves developed and put out there, free for academic use, just saying. Yeah, and so I think if you go for a much cleaner look like that, where the rendering is physically accurate, the shadows are much less harsh, the entire protein just becomes a little bit easier to look at, and it's easier to bring across what the part of the protein is that you'd like the viewer to have a look at. it's been mentioned a few times already, kind of the problems that you run into when you've got something in 3D and you need to show it as a flat 2D image. We've heard about the Coot shake and all those sorts of things. And this is something I could really recommend that you look into for your papers, is just stick animations in your supplementary. And I guess the big difference in this kind of approach compared to the kind of turntables that you see that you can get out of PyMOL or wherever, is that here we're animating the camera. And I think that's a much clearer way to show things, because you've got the full view of the entire protein, and then you're slowly going into your region of interest. And so that is much more understandable if you're not exactly comfortable with protein structures as the sort of more general scientific audience. And of course, then showing dynamic processes, that's something where, as you can imagine, animation really comes to the fore. And I've got some work in progress here that we're working at the moment for Jan Leuwen's lab at the LMB. This is a structure solved by Nicolas Jean. It's the FTSK DNA motor. And what Nicolas has done is that he's solved all the different conformations as this motor is kind of gobbling up DNA. And we've animated that. This is, off the top of my head, it's like 1 in 5,000 speed or something like that. And the great thing about this is that nothing here is kind of hand manipulated, hand animated by us. It's as opposed to that kinase that you saw in the showreel, where we actually had to hand animate how that's walking around. This is just interpolating between the different conformations that they solved. And the nice thing, where if you go into these kinds of animation software, is that you get a little bit of freedom with how you represent things. And so in this case, one of the issues that they ran into was the fact that it's quite an intricate combination of different conformations. And in this hexamer, there are 6 different conformations, and it's sort of treadmilling round. And we were talking about different ways that we could emphasize that treadmilling effect. And so here, I've gotten rid of 2 out of the 6 and color-coded each particular conformer. And so you can really see that that color is treadmilling around the hexamer. And then on top of that, in red, you see the specific residues that are contacting the DNA, and it's quite a nice effect that you can see them sort of push the DNA through. So that's the kind of more direct way of showing how the data looks. And then there's what I'd like to call the editorial way, and that's more the type of thing that you would send in for your journal cover submission. So you get a little bit more artistic license here in how you approach things, and there you can do some interesting things as well. And so here, this is a structure that we visualized for Laurie Passmore at the LMB. It's the Fanconi anemia core complex. And in this case, we didn't have to do a lot of fancy things, because the complex itself is really quite pretty, and you just want to make it as shiny and as lovely as you can. And so we used some very dramatic materials here, where you get that kind of translucency like you see on the left there. The other thing that you can do with animation or animation software that's quite nice is you can really start building up scale. And so here you're looking at histones, and in this image combined, you've got around 1 million base pairs. And it's always something that's, particularly when you're communicating to the general public, conveying this sense of scale and just how busy things are inside of a cell is always quite difficult to do. And so these types of approaches, I think, are really quite useful. And then I've got a couple of images here that we did for Elton Zekeraj in Leeds that kind of explored different themes as well. And so here, what you're looking at is a complex between a deubiquitinase and a pseudo-deubiquitinase. And we're trying to grappling with the idea of, like, how do you show the differences between them, because they look very, very similar. How can you show this idea that one is catalytically active and the other one isn't? And so what we decided on in the end is this idea that the active deubiquitinase is really ordered, and it's built up of these really nicely, neatly stacked Lego blocks. And the pseudo-deubiquitinase that's not catalytically active is really chaotic, and is this jumble of little cubes. And another thing that we did for Elton was around his recent complex between BRISC and SHMT2, and that's a complex between 2 sets of proteins that really, on the face of it shouldn't be formula complex, and we wanted to make this image to convey the idea that there's connectivity where it's not expected. And so we went for this kind of plexus-like integrative approach, which is a bit unusual in how you would otherwise show your proteins, but then that's the artistic license that you can sometimes adopt. So I wanted to kind of take you through the steps and some of the thinking that goes in going from something relatively simple to a much more complex final image. And this is some work that we're currently doing for Helen Walden up in Scotland. And what you're looking at here as a sort of a starting image is FANCD2 in the bottom, and it's a story about the ubiquitination of FANCD2. And in the top right-hand corner, you can see the E2 ligase UB2T. And the entire point of this paper, I put in print. I had a look just now, actually, Christmas Eve. They put it up online already, so it's out there. The story of the paper is how basically UB2T is able to recognize one specific lysine out of the dozens of lysines on FANCD2. And the important bit is the fact that you get two triads that you can see in orange and in blue there that are coordinating. So this is kind of your starting point, and it's something that's quite clean, and you could even imagine something like this in a bit more of an academic setting rather than the editorial side of things. So the first thing that we did is to make this a lot more dramatic. And this is not a very contrasty screen. So I'll go through this a little bit more quickly because, yeah, it's going to be difficult to show in great detail on the projector here. But the idea is you go for something a little bit darker and you add some glow effects and you add some light effects. So you've got your important triads and you make them emit light, and so you get this lovely kind of cast light in orange and in blue up there. In order to grab the attention of the viewer and put it a little bit more into the foreground, you can add some fog effects in the background to kind of make it obscure back off in the distance. And then we were talking about this concept of UB2T scanning across the surface of FANCD2, and Helen had as a comparison the idea of like having a submarine go across the ocean floor with a little searchlight and trying to look for things there. And so that searchlight worked quite well. And if you then add additional glow effects onto that and some more glow effects and some more color correction, then you can sort of start layering on to these things. And if you then kind of flick back and forth between your original image and your heavily color corrected image, you can see that that's something that you can make much, much more dramatic from your initial render. And it's also definitely something to keep an eye on social media, because if you see it on the screen, it's quite a bit more dramatic. Software. When it comes to animation, people get just as invested in their choice of software as they do with structural biology, and I think Kevin really hit the nail on the head yesterday, talking about not which is best, but kind of why the different packages differ, what you can do with different packages, and why you might be interested in having a look at them. So there's three packages that I briefly wanted to discuss. The first one was already mentioned earlier today, is Blender. Blender has come a long way in recent years. It used to be quite basic, but now it can do loads and loads of really cool stuff. The thing is, though, Blender is not visual effects industry standard. If you have no ambition to kind of pivot into animation and maybe go and work with an animation company in the future, then that's absolutely fine. But it's something to know. If this is something you're interested in, if you go and work with an animation company, you will be asked to learn something other than Blender, because it's just not being used as industry standard. It's free, though, so that's definitely a very big bonus point there for Blender. And there's a plugin called BioBlender which allows you to import structures into Blender and visualize them. So our main software that we use is Autodesk Maya. That is kind of the industry workhorse in the visual effects industry. It's usually quite expensive, but amazingly, it's free for academic use. So as long as you still have that university email address, use it and get it for free. And I think it's free on a 3 or 4 year license or something like that. There's two different plugins that you can use to get PDBs into Maya. The first one is Molecular Maya by Gail McGill at Harvard. The second one is EPMV, done by the Olson Lab at the Scripps. The problem with those is that you're, with both BioBlender and with Molecular Maya and with EPMV, you're reliant on these external plugins to get your proteins into the software. And it can run into compatibility issues, and we've had it happen before that a new version of Maya can do amazing things. We pivoted to the new version, and then all of a sudden Molecular Maya didn't work anymore, and for about 3 months we were waiting for updates and couldn't actually get our proteins imported in the way that we wanted. So yeah, there's a bit of a dependency there on plugins that can be unfortunate, which is why Houdini is the software package that I would heartily recommend to you. So this is also a VFX industry standard package, but it's for a very particular goal, and that's if you need to do something that's difficult. So Houdini is very good at large-scale particle simulations, particle physics stuff. So in Hollywood, people use it whenever they need to blow something up, whenever they've got splashes, where they've got smoke, when they've got sparks, all that sort of thing. Houdini is amazing at that. Again, it's free for academic use. So that's another bonus point. And the thing that really sets it apart is that it natively imports PDB files. Unfortunately, not MMSIF files, but PDB files it can handle. So the really big advantage about the fact that you can natively read PDB files in the software is that with the other ones, when you import a PDB, and you're looking at your structure and you've set your initial parameters of how thick you want the ribbon to be or how blobby you want the surface to be, that sort of thing. It's an object that can then no longer change. So it's the things that are really easy to do in PyMOL, like selecting a particular residue and making it red, you can then no longer do unless you want to go in and manually select the polygons that correspond to a particular residue. So it's quite quite a headache, really, to do that sort of thing that should be really, really easy. With Houdini, because it natively reads it, you've got all of that information. And so here's a quick look at the interface. And as you can see on the left, it imports a structure as a point cloud. And so you've got it right there. You've got your coordinates. And it's amazing because then you actually see some of the data. And you won't be able to see it from far away, but on the left is all the data that's in your PDB file. So you've got your sequence, you've got your residues, you've got your B factor, you have all of that stuff right there in your animation software that you can then go and manipulate and play around with. And one of the things that makes a lot of visual effects artists quite afraid of Houdini is that in Houdini, every now and again, you need to write 1, maybe 2 lines of code. And visual effects artists are terrified of writing code, but something tells me that a lot of you aren't. So that's definitely something to keep in mind. So I put together a little kind of structure-specific showreel for you to have a look at the type of thing that you can directly do in Houdini in different types of representations of structures. So there was a lot going on there. If we go through it at a slightly slower pace, obviously you can just trace a line through all of your alpha carbons and you can get a ribbon representation. You can show your side chains, and the nice thing, like I said, because you've got all that additional information in there, you can color them according to whatever you desire. And so in this case, I colored them based on their B factor. I put a whole bunch of turbulence into the scene. I scaled the effect of that turbulence based on the B factor, and then you get a nice little wiggle that's wiggling in all the areas that you would expect based on looking at the structure. And so the nice thing about this is it means that you can start showing proteins in animations in a less static, more dynamic way, and it's something that we know that these things are dynamic in the cell, and it always pains me a little bit to have to animate them as a static thing floating around and going towards the membrane or wherever. You can do surfaces, you can tweak all kinds of parameters to determine how kind of cartoony and blobby you want it, or how restrained you want this. And then I usually always quite like a low polygon kind of representation, and it's not just as a sort of stylistic approach. If you're filling a scene with loads and loads and loads of proteins, then going for something that's slightly lower poly in the background where you can blur it away, you don't see the angles, means that you've got a bit more computational leverage to really up the protein counts in a cell. You can get away with thousands and thousands of proteins in your image. And then, of course, you've got conformational changes. And so the nice thing is if you've got your two different structures that have roughly the same amount of missing data that corresponds to one another, you can quite nicely integrate between the two of them. And when you show a drug flying in, you can actually interpolate and get that conformational change, which is also what I was showing you in the work we're doing for Jan Leuwe with the DNA motor. So I'd say watch this space. We're currently working on putting a bunch of tutorials together, putting some example files together, because, like I said, all of this software is free to use if you're academic, and you know, sadly, I can no longer use that excuse, and I'd very much recommend you use it as while you have the logon addresses for that. Yeah, and so as soon as you've got these kind of example files, it's quite easy to, once you've got everything built up, it's easy to swap out one protein for another, and it's quite efficient that way. So in terms of acknowledgements, I have to give a really big thank you to Peter Parker at the Crick, my PhD supervisor, who had the patience to let me kind of learn all of this sort of thing on the side while I was doing my PhD with him. And then throughout the presentation, you've seen work that we've done for Neil MacDonald, Richard Treisman, and Charlie Swanton at the Crick. You've seen stuff we've done for Alessandro Vanini at the ICR, for Jan Leuwe and Laurie Passmore at the LMB, Helen Waldman in Glasgow, Elton Zekaraj in Leeds, for Claire and Patrick Ayers in Liverpool. I'd like to give a particular thank you to Helen Waldman and Jan Leuwe for allowing me to show some ongoing work that's not quite finished yet. And Tagma, if you want to give Houdini a go, and Tagma are the people that make the most fantastic tutorials, and Moritz and Manuel are really helpful as well if you run into trouble to help troubleshoot things for you. And of course, I'd like to thank the organizations for this wonderful meeting. Thank you. And of course, if you do want to try these things out and you run into issues, that's my contact details. Feel free to drop me a line and I'll be happy to troubleshoot.
Right, that was wonderful. Thank you very much. And well, thank you for accepting the invitation in the first place. Right, boss, do we have time for a couple of questions? Okay. Who wants to ask? Andy.
Thanks, that was great. One of the things that struck me is, especially in the early parts, is the way that you make use of limited depth of focus.
Yes.
Which I don't think we use very much because I don't know of any tools that do that. So which of these tools will do that kind of thing?
Everything, basically. So when you, with any kind of visual effect, or animation package, visual effects package, the way it works is that you get a camera in 3D space. So that's why you could animate that camera like I showed you. But that camera, any type of parameter that you can tweak with a normal physical camera in real life, you can tweak there. So it's not just about depth of field, which you can tweak, but also what type of lens you're using. And so you can, if you want to, you can use a wide angle lens to get more in the field of view. But then obviously if you, like you do with wide angle lenses, if you get quite close to the protein, it looks distorted and weird. But this is, yeah, it's something that's universally available in all of these packages. And I quite like depth of field, not just because it gives this sort of filmic look, but it really helps guide someone's attention to a particular part, and it gives you that sense of depth that helps in a 2D image as well. Yeah, thank you.
If you wanted to get started with this, what kind of computational firepower do you need?
So in terms of rendering everything is, or most things are CPU based nowadays. The shift is happening to GPU based rendering, but it's a slow change because some of these, like the Pixar software has been knocking around since the late 80s, and so it takes a bit of time to convert all of that. So it depends on how long you want to wait, really, because you can run things on a laptop. It's just the rendering is a bit slower. But one of the things that, the workstation that I'm using for most tasks is quite high spec, but to be honest, as soon as it comes to rendering, you can do all of that stuff over on the Google Cloud. Google Cloud have a very particularly dedicated cloud rendering service. And the nice thing there is you can use 50 machines at the same time, up to 64 cores per machine. And so that's the type of computing power that as a small studio, you could never aspire to have yourself. And it's quite reasonably priced as well for the computing power there. So that's what I'd recommend. You optimize things in low resolution on your laptop or whatever it is you have. And then for the final resolution, you send that off to the cloud.
Yes, maybe a slightly facetious question. You're obviously at the upper end of biomolecular graphics, and seeing your background and the materials you work from, you would never make that sort of mistake. But why is it that one constantly comes across all these company logos or conference flyers with left-handed alpha helices and DNA helices? I'm
a stickler for the old left-handed DNA. I hate seeing that. I don't know. There's an example. I don't have the slide on me here, but there's in the medical animation community, there's a very famous example where University of Southern California had a big donation to build a new neuroscience institute, and to celebrate this, I think $60 million donation, they took out nationwide newspapers, full-page ads, and the brain was back to front in that image. And it's just the most embarrassing thing. And so this is why I always say if you want to get an animation done, hire medical animators and not sort of a general animation company that doesn't understand anything about what it is that they're doing when it comes to science. But yeah.
It seems that whenever pictures of molecules are put in the hands of graphical artists, there's a good chance that they will be flipped over with the wrong hand. Yeah,
and it's even in things like Jurassic Park that the DNA is left-handed, and yeah, it completely pulls me out of the movie when I watch that sort of stuff. Yeah.
Okay. Thanks.
Thank you.
Another question? No? Right. So I think this has come to an end. So please join me in thanking Jeroen.
Jeroen, yeah.